Everybody's ready? We are going to be having questions from every single lab that we've had, including today. So first, we're noting our tic-tac-toe board. We need to be able to identify the right hypochondriac, epigastric, left hypochondriac. We got to get our left and right correct. Our umbilical, and then we would note the right inguinal or iliac versus the hypogastric and the left inguinal or iliac. Don't expect to see exactly the same box over there, okay? Then we're looking to identify the different planes. So remember, if we're going left to right, it's sagittal. Front to back, coronal. Top to bottom, transverse or horizontal. And if we're at an angle, it's inguinal. We'll be identifying major areas that are kind of common, starting with, think in terms of the eye. So we're talking about orbital, cervical, axillary, inguinal. The back of the knee is popliteal. What do we call the pelvic floor? The perineum or perineal region. Identifying the actual serous membrane cavities, not necessarily the actual parietal versus visceral layer. So first, it's noting the mediastinum, the open area in the middle behind the sternum, and then our pleural cavity, our pericardial cavity, and our peritoneal cavity. Spelling counts. So remember, there's a perineum versus the peritoneum. Okay? Peritoneal cavity. Now we're going to have a few stations with the cell model. That's why I put two images here. We're going to be needing to not just identify the organelle, but also recognize their function. So one station may say nucleolus, the next station it's the nucleolus again, but it's asking what's its function. So we'll go by the nucleus, concentration of chromatin, direct cell function. The nucleolus, messenger RNA, and directs protein synthesis. Mitochondria, the folds are called cristae. It's the oxidation of glucose, production of ATP. In other words, a very brief description of what you can squeeze in on the line. Okay? Even if you put down ATP, that works for me. Okay? Then we get to smooth ER, lipids, and detoxing. The rough ER and the white dots are ribosomes, polypeptide synthesis. The Golgi, complete protein synthesis. And then we go to the centrosome, the microtubular organizing center, MTOC. And then centrioles, they make the spindle for mitosis. And then we would just note a lysosome. The lysosome is a storage vessel. So again, that's why you see I have two images here. We're going to be taking the images that we see here of the model of mitosis, and they're going to be placed in out of sequence. And your responsibility will be to put a letter answer that indicates the correct sequence. So we note up here we're in interphase, early prophase, and we're only going to have one image of prophase, metaphase, anaphase, and then telophase with cytokinesis, and you'll be asked to identify the cleavage furrow and then describe the process of being cytokinesis. If we look at the one on top just for a minute and if let's say the lettering here is A, B, C, D, E, yeah, well then, that would be the correct sequence. But if let's say this was A, this was B, and this was C, and this was D, then your answer would begin with D as being the correct sequence. All right, that's how you're going to be doing that. You're not going to be writing out interface, prophase, metaphase. You're going to be saying what's the matching of the models. And while we're matching up the models, you'll be asked to identify on, I don't have the image here, it was the image of the